Cell cycle. The cell cycle has three states, which are quiescent, interphase, and mitosis, and five phases, G0, G1, S, G2, and M. The quiescent state, also known as the senescent state, comprises of the G0 phase. Examples: neurons, cardiac muscle, and RBCs. Interphase state consists of G1, S, and G2 phases. And the mitosis state consists of the M phase. Prophase, metaphase, anaphase, telophase occur during this phase. The three major cell cycle checkpoints are the end of G1, the end of G2, and during metaphase of the M phase. Progression through checkpoints is regulated by cyclins, cyclin-dependent kinases, and tumor suppressors. Cell cycle progression is controlled by a variety of different cellular mechanisms, such as tumor suppressors, genes that code for transcription factors, proteins that detect DNA damage, enzymes that allow the cell to progress to the S phase, and signaling molecules that activate those enzymes. F regulates expression of transcription genes and cyclin-dependent kinase 2, cyclin E, and cyclin A. If there is significant DNA damage, P53 stimulates production of P21. P21 binds and inhibits all cyclin CDK complexes, which leads to arrest of cell cycle until the DNA damage is repaired and P21 levels drop. Blastoma protein inhibits E2F expression. Cyclin D CDK4 and cyclin D CDK6 complexes phosphorylate retinoblastoma protein that leads to inactivation of retinoblastoma protein and expression of E2F. The expression of E2F then leads to the expression of transcription genes and formation of cyclin E CDK2. Therefore, the cell is pushed into the S phase. Cell cycle regulation in S and M phase. Cyclin A CDK2 is required for DNA synthesis. Cyclin A CDK1 and cyclin B CDK1 promote the events of mitosis. Towards the end of mitosis, the anaphase promoting complex causes ubiquitination and destruction of cyclin A CDK1 and cyclin B CDK1 that leads to termination of M phase with anaphase, telophase, and cytokinesis. The most radiosensitive phases are the G2 phase and mitosis. The least radiosensitive phase is the latter part of the S phase.